Hello everyone. Today we are going to discuss one of the applications of biotechnology, that is gene therapy. Now in the year 1976, a television movie by the name of The Boy in the Plastic Bubble was released. And again in the year 2001, another movie by the name of Bubble Boy was released. Now in both these movies, the boy was kept inside a plastic bubble. This was done to protect the boy from the various diseases caused by the germs present in the normal air. This boy had to be kept isolated away from the crowd because he could not breathe the normal air. He had to be supplied with the clean air which was free of germs. The boys present in both the above movies, they were suffering with the same genetic disorder which caused the collapse of their immune system. The genetic disorder was due to presence of a defective gene which was not able to code for the enzyme ADA, that is adenosine deaminase, which is responsible for the proper functioning of the T lymphocytes. Now let's go to the history of gene therapy. Father of gene therapy is William French Anderson. The first gene therapy was performed in September of the year 1990. First patient who obtained it is a 4-year-old girl by the name of Ashanti De Silva, daughter of Raj De Silva and Van De Silva. They belong to a place known as Bethesda, Maryland in USA. After the birth of this child continuously was suffering with different kind of diseases, and by the year 1989, the parents had lost all the hopes of her survival. But in the year 1990, they connected with the American physician, geneticist and the molecular biologist Mr. William French Anderson, who was lobbying for permission to move forward with human gene therapy trials. Gene therapy is a kind of treatment of different kind of hereditary disorders which are caused in our bodies. These hereditary disorders, they are caused because of the presence of a single defective gene in our cells, and sometimes due to the absence of the gene in our cells. So what is done in gene therapy is remove the defective gene from the cells and insert a functional gene in its place. Now after the insertion of the functional gene, this gene will start giving the proper orders and the cells will start functioning properly and the person becomes cured. So we can also say that gene therapy acts like a medicine in certain cases. Genes which are present on our DNAs, they have a specific code for the formation of a specific protein, and that specific protein once produced, it dictates a specific cell function. So all this takes place by a process known as central dogma. The gene present on the DNA undergoes transcription to produce RNA, and this RNA undergoes a process known as translation and produces the proteins, and once the proteins are produced, they will control a specific body feature or a specific body function. In gene therapy, certain viruses known as retroviruses are used as a vector to transfer the functional gene into the cells to replace the defective ones. Now these retroviruses have RNAs as their genetic material. So during this process, the RNA present in the retrovirus undergoes a kind of transcription which is known as reverse transcription, by which method it produces a DNA known as the complementary DNA. This DNA now undergoes transcription to produce RNA. The RNA will undergo translation to produce proteins, and now these proteins once produced, they can control a body feature. Many of the proteins, they are converted into different kinds of enzymes also. Here we should remember one thing, that all the enzymes are proteins, but all the proteins are not enzymes. We are taking the example of SCID to discuss about gene therapy. The full form of SCID is severe combined immunodeficiency disease. It is also known as adenosine deaminase deficiency disease, or it is also known as the bubble boy disease. SCID is a genetic defect in which the infants lack antibody formation as the T lymphocytes are not functioning properly in the absence of the enzyme adenosine deaminase. Okay, now children suffering with SCID, they can die even of ordinary diseases, vaccinations and blood transfusions. 25% of the SCID patients, they are born with a defective autosomal gene for the synthesis of the enzyme adenosine deaminase, and many of the patients, they do not have the gene which is responsible for the formation of ADA enzyme. Now, patients suffering with SCID, they are treated by three methods. The first method that is mentioned here, that is the bone marrow transplantation or the BMT method. The bone marrow transplantation method requires a donor, a healthy donor which should be a very close family member or a very close relative of the patient. The healthy bone marrow cells of the donor, they are taken and these cells are implanted in the bone marrow of the patient. Now, once the healthy cells are implanted in the bone marrow, the functional gene present in these cells, it will start guiding the formation of the ADA enzyme. When the ADA enzymes will be produced, this will stimulate the T lymphocytes and make them functional. The T lymphocytes start producing antibodies and the patient recovers. But this process, bone marrow transplantation, the patient's survival rate is very less. The patient may survive for 1 year, 2 years or a few years only. Second method to treat SCID is enzyme replacement therapy. In this method, enzymes which are manufactured in the laboratory conditions, they are injected in the body of the patient. These enzymes start working immediately and the antibody production starts. The patient recovers. But these enzymes cannot survive in the body for very long time. So, the enzyme injection has to be given again and again. And the third and the best method to treat SCID is the gene therapy. Gene therapy where introducing a functional ADA gene into the lymphocytes is done. Gene therapy uses patient's own cells, so there is no risk of graft versus host diseases. Gene therapy is of two types. First type, that is the somatic gene therapy, where the introduction of functional gene is done into the somatic cells of the patient to replace the defective gene. Second type is the germline therapy. Here the functional gene is introduced into the germ cells of the patient, such as the sperm, the ovum or the zygote. But this germline therapy, it is at present under trial on the farm animals only. Due to ethical issues, till now it has not been tried on the humans. Now let's go through the steps of gene therapy. Step 1 is isolation of desired gene. This desired gene can be isolated from the body of or from the cells of a healthy person, and this desired gene, it has the capability of producing the ADA enzyme. This gene can also be created under a lab conditions. Second step is amplification of desired gene. Okay, now this desired gene I have shown here in green color. This gene has to be multiplied many times because we require a large number of this desired gene, and this can be done by the method called PCR, that is polymerase chain reaction. This is also done under the lab conditions. Step 3 is preparing a therapeutic vector or a retrovirus which can carry this desired gene where we need it to send. For preparing this retrovirus, which is also known as a therapeutic vector, first the retrovirus is taken. Inside this retrovirus, because we have already discussed, it is having RNA as its genetic material. This RNA has a viral gene. This viral gene, which is responsible for causing a disease when it infects a person. So this viral gene has to be removed. Okay, so after the removal of this viral gene from the RNA of this retrovirus, now this virus is known as genetically disabled retrovirus. Okay, because it lacks the viral gene for causing diseases. Now to this genetically disabled virus, we have to insert the desired gene. The gene which we had amplified here, a large number of retroviruses are taken which are genetically disabled, and these functional genes are inserted in the or inoculated in the RNA of these genetically disabled retrovirus. After the inoculation of the functional gene in the RNA of the retrovirus, now this RNA has become a recombinant RNA. That means this virus is now carrying a recombinant RNA with the functional gene incorporated in the RNA of this retrovirus. This is now known as the therapeutic vector. Step 4 involves involvement of helper viruses. The viral gene of these helper viruses are also removed. These viruses provide viral structural protein for generation of more vectors. Step 5 is to determine the eligibility of the patient, that is, accurate diagnosis of the genetic defect. This is done by the use of a DNA probe. And step 6, this is delivering the working gene to the patient. Now we will discuss how to deliver this working gene in the body of the patient. We are talking about the SCID patient, students, and this hand represents the body of the patient. Step 1 is to remove lymphocyte cells from the blood of the patient. These lymphocyte cells, they have cellular DNA with the defective gene. That means these cells, they are not able to produce the adenosine deaminase enzyme. These cells are put in a culture medium to grow them under the lab conditions. Step 2 involves infection of these cells with the prepared retroviruses. These retroviruses we had already prepared ex vivo. Whatever processes are done under the lab condition, that is known as ex vivo. These retroviruses or the therapeutic viruses, they contain the recombinant RNA which is having the functional gene for the production of the enzyme ADA. Now these retroviruses are brought in contact with the lymphocyte cells which we extracted from the body of the patient. Now viruses have the power to infect the living cells of the organisms. So when these two are brought together, this virus infects the lymphocyte cells. During this infection, the recombinant RNA of the virus enters inside the body of the lymphocyte cell and comes to lie in the cytoplasm of the lymphocyte cell. Now, the protein coat of the viruses never enters the cell. It is always the genetic material that they contain. This recombinant RNA of the virus present inside the cytoplasm of the lymphocyte cell now undergoes a process of transcription. This transcription is known as reverse transcription here because from a single stranded RNA, now a double stranded DNA is produced, and this double stranded DNA here, it is known as the complementary DNA and represented by small c and capital DNA. This DNA is having the functional gene which is responsible for the formation of ADA enzyme. This complementary DNA enters the nucleus of the lymphocyte cell and it becomes mixed up or it becomes incorporated in the DNA of the lymphocyte cell. Now we can see that the lymphocyte cell has a recombinant DNA. Now step 3 was the formation of the recombinant DNA inside the lymphocyte cell, and this DNA, it is having the normal gene. This DNA is now having the normal gene which has become incorporated into the genome of this lymphocyte cell. This is also known as the genetically engineered cell, and step 4 involves return of this genetically engineered cell back into the blood of the patient's body. Now the lymphocytes are present inside the blood of the patient. These lymphocytes, which are a kind of white blood cells, and especially the T lymphocytes, which are having the functional gene now, they start producing the enzyme ADA, that is adenosine deaminase enzyme. Once these enzymes are produced, they make the T lymphocytes functional. Okay, the T lymphocytes were not functional due to the presence of the defective gene, but now they become functional and hence they start producing the antibodies. So once the lymphocytes start producing the antibodies, the immune system of the person becomes stronger and that patient survives. Now the survival rate here depends. Now the T lymphocyte cells or these white blood cells, these T lymphocyte cells have somewhat longer life when compared to the other blood cells, but they are not immortal. So this process of gene therapy, it has to be repeated in the patients because the lymphocytes are not immortal. Similarly, the bone marrow transplant which we discussed, this also After one transplantation, the patient can survive for a few years, but again those cells are not immortal and this also has to be repeated. However, if the gene isolated from a normal person could be introduced in the bone marrow at an early embryonic stage, then a permanent cure can be achieved. But due to ethical reasons, it has not been done. It is not possible to know whether a child is going to suffer with a genetic disease, because in many cases the parents are healthy, and many times mutations in the healthy persons or healthy parents in their reproductive cells might be the reason for the occurrence of SCID in their children. Finally, we will go through the list of diseases which could be treated by gene therapy. Some have become successful, but some are still in clinical trials. So diseases like SCID we already discussed, cancers like leukemia, lymphoma, melanoma, prostate, breast cancer, etc. A disease like thalassemia, muscular dystrophy, atherosclerosis, cystic fibrosis, hemophilia, rheumatoid arthritis, peripheral vascular disease, AIDS, and many more diseases are there, genetic diseases, which could be treated by gene therapy. So scientists are still doing the research. All the best to humanity. Let us hope we become successful.